My name is Karan, Bhatia. I'm a solutions architect with Google, and I lead our efforts around scientific computing. And I'm here with a distinguished set of panelists, Matteo Chinazzi, Salvador Dura Bernal, Ben Torkian, Paul Sagona, and what we have for you today is a panel session about how people are leveraging Google Cloud for doing scientific computing and for research computing. It's really a great time for scientific computing. It's really exciting to be a computational scientist today. The access to new processors, new accelerators, and new software frameworks are really enabling the researchers to harness a large amount of compute capability and really putting it at their fingertips to advance science. Google Cloud is a worldwide system. We have built it on a global network that includes everything from the fiber optic cables to the servers to data storage. We have 17 regions distributed around the world. We have thousands of point of presence sites, undersea cables and submarines, and we have sharks that bite. You might have heard that we have sharks that bite the fiber optic cables, and we have to go down and fix it. So all of that is owned by Google, and that capability with 17 regions around the world is really made available to everybody, including the academic research community. So some of the things that we do for the scientific community. So the first thing is that we have a pretty robust set, a free tier of services for our products. So not only do you get, when you sign up, you get, I think, about $300 in credits to use generally, but most of the products themselves have a free tier. So for example, if you're using BigQuery, you can use a certain amount of BigQuery every single month, a certain amount of GKE and other services. Every service has a free tier, and that's a great way of exploring and using and learning about the Google Cloud Platform for your research. If you need more than that, we have a research credits program where we can provide some more credits for you to leverage Google Cloud for your research needs. And we're working very extensively with the funding agencies to make it really easy to allow you to get funding, get Google Cloud credits for your research, your multi-year research, your research projects. We've had a really strong collaboration with the National Science Foundation, and we continue that with building more programs and more interactions there. And earlier this week, we announced a collaboration with a partnership with the National Institutes of Health as well. So across the board, we're really making it as easy as possible for scientific and research computing to be done with Google Cloud. And to show you guys what people have done with this, please, before we bring the speakers up, there was one more thing. There's a National Science Foundation report that just came out that talked about this. There was a workshop in January that brought together a lot of academics. This was run by Jennifer Rexford in Princeton University. And it talked about how to facilitate Google Cloud for academic use and why to do that. So there's a number of recommendations there within that report for universities and for academic departments. And I encourage you guys to take a look at that. So please join me in welcoming Matteo to talk about his research and how he's leveraging the Google Cloud for his science.
Hello, everybody. Thank you, Karan, for the introduction, and it's great to be here. So I would like to start my presentation off by asking you one simple question. What will you do if somebody came to you and asked you to model mosquito bites? Well, that was our task back in 2016, and in this presentation, I'm going to tell you how we developed a model with the help of GCP to study the diffusion of Zika virus across the globe. And just to give you some context, so back in 2015, Brazil had notified the World Health Organization that a new mysterious virus was circulating in the country. And a few months later, that virus was recognized to be Zika. And moreover, during the same year, they also noticed an unusual increase in the number of microcephaly cases in the country. And for the ones of you that are not familiar with microcephaly, it is a rare neurological condition in which infants' heads are significantly smaller than the ones of other kids of the same age and sex. And it may cause not only physical but also learning disabilities as newborns grow. So because of this, there was soon suspected that a link existed between the appearance of Zika virus in Brazil and microcephaly and other congenital and neurological conditions. And for this reason, the WHO declared in February 2016 a public health emergency of international concern. And that's actually why we decided to start working into this project. So our goal was essentially to try to develop a model that could help public health officials to better understand the evolution and the dynamic of the Zika outbreak across the world. And by the time we were done, not only we have done just that, but actually we have developed a new platform, a new framework that could allow us to study also other diseases, as I will tell you later on at the end of this presentation. So now let me tell you how we did it. In the next few minutes, I'm going to talk to you about three things. First, I'm going to tell you what were the scientific and computational challenges that we faced and how we overcame them. Then I'm going to tell you what are the main lessons that we learned while using GCP in our journey. And at the end, I will give you a taste of the kind of scientific results that we can obtain using our model. But before diving into the scientific challenges, let me take a step back and give you a piece of good news. So luckily for us at our labs, and thanks also to our network of collaborators, we have access to a team of experts that know how to model contagious outbreaks. And in particular, at MOB's lab, we have a long experience in modeling anything from the seasonal flu to Ebola. And actually, one of the things that we do best is to develop computational and analytical models to study the spatial spread of diseases. And at the core, at the foundation of many of our projects, we have GLEAM, which is our global epidemic and mobility model that essentially allows us to simulate and track the evolution of an epidemic across the world from beginning to end. So let me just spend 1 minute telling you how GLEAM works. GLEAM essentially is a piece of software that allows you to combine different data sources, high-resolution census data, mobility data, and complex disease dynamics into a unified framework. And the way it works is that it makes use of this high-resolution population data to divide the world into different subpopulations, which are then connected to each other through a mobility network that, for example, take into account for the fact that people daily commute to go to work or take airplanes to travel. And then what the model does is that it takes each subpopulation and simulates the diffusion of a virus within that subpopulation while taking into account for all the possible interconnections that exist between them because of the mobility network. So essentially what GLEAM allows us to do is to simulate the evolution of an epidemic at the global scale. Unfortunately, though, for us, when we started this project, there was one thing that GLEAM was not able to do, and that was model mosquito bites. So the first thing that we had to do was to introduce, first of all, a mathematical model that could allow us to model mosquitoes within GLEAM. And second, we had to introduce additional data sources within the computational framework of GLEAM actually to indeed model mosquito presence and biting patterns. And the reason why we had to do that is because the main drivers of Zika transmissions are infected mosquitoes that by biting individuals inject the virus within a population. And then those same individuals, in turn, when they are bitten by otherwise healthy mosquitoes, they can help propagate the virus back to the mosquitoes, and then the cycle goes on. So, however, not only we had to, as I said, add a new mathematical model within GLEAM, but we also had to add new data sources together with the population and mobility data we already had to account for different things. The first one is that we had to introduce very detailed mosquito maps telling us where around the world there are mosquitoes that are able to actually carry Zika virus. Then we also had to add monthly varying temperature data to actually model mosquito life cycles, so mosquito lives and deaths. And then on top of that, we also had to add socioeconomic indicators to account for the fact that if you live in an area where you have access to better sanitation facilities, or simply there is a more widespread use of air conditioning, then you are less subject to be bitten by a mosquito and therefore of contracting Zika. After we have done all of this, however, we were not done because there were other challenges that we had to face. First and foremost, we did not know the original time and place of introduction of Zika virus in Brazil. And that essentially meant that we did not know from where and when to start our simulations from. Plus, since now we have additional complexity in our model, we have many more additional parameters that we need to calibrate, because not only we have to trace and simulate people movements, but also mosquito presence and biting patterns. And obviously, since it was the first time we were doing something like this, we also would like to have a computational framework and pipeline that will allow us to have quick iterations between different versions of our model. So all those problems actually translate pretty directly in some very specific computational challenges that we had to address. The first one is that since we did not know the original time of introduction of the virus in Brazil, we had to run longer simulations with GLEAM to essentially explore back in the past what was going on. And that meant that each single simulation of a global epidemic will take up to 7 minutes on average to run. However, because of the randomness that we have and the complexity that we have in the model, we actually have to run 200,000 of those simulations to have robust results. And that, unfortunately for us, generates about 25 terabytes of data that then we have to process. And as I said, we would like to do all of this, run the simulations, and analyze the results quickly, meaning in less of a day. And that's essentially where GCP came in to save the day and help us with our research. So what is the cloud-based solution that we devised to address those challenges? First of all, our first stop was actually to deploy GLEAM on Compute Engine to essentially help us scale up in terms of number of parallel jobs we could run at the same time. And to give you an idea, that meant that at peak research hours, we were using up to 30,000 cores at the time. Moreover, since we did not want to actually saturate our on-prem NFS partitions, we decided to store from the very beginning all our data and raw data on cloud storage, and to use the power of BigQuery to actually parse all those terabytes of data that we were generating, to have essentially all our simulations done and analysis within a day. And then our pipeline will close, essentially moving back all the process data to our local machines, where we will generate then the figures and the numbers we wanted to look at to analyze what happened with our simulations. But let me give you some numbers to get an understanding of the scale of our efforts. So overall, we simulated more than 10 million global epidemics from beginning to end. And that amounted to more actually than 250 compute years. And to run all those simulations, we deployed more than 450,000 instances on the compute engine and analyzed more than 300 terabytes of data on BigQuery. While doing this, we learned a few things. First off, we learned that in order to save money, actually was better to move from standard VMs to preemptibles, because that essentially allowed us to run 5 simulations for the price of 1, and to profile GLEAM, so to profile our software, for example, in terms of memory usage, to then allow us to use custom machines instead of regular instances. And in terms of time saving, what we found was that it was extremely beneficial to actually run many concurrent loading jobs in BigQuery to drastically reduce the amount of time we had to wait to actually populate the database with all the terabytes of data that we were generating. And last but not least, we automatized our entire pipeline using the GCP client library for Python to allow us essentially to run all the simulations plus all the analysis and get at the very end a very nice PDF report with all the numbers and figures we wanted to look at without human interventions. So now let me give you a taste of the kind of results and the kind of things we can do with this. So first of all, we can use GLEAM as our own personal time machine to go back in the past and try to figure out what was the original time and place of introduction of the virus in Brazil. And for example, what you see there is that we find that the most probable time of introduction was between September 2013 and February 2014. But of course, what we can do is that we can also peek into the future. So what we can do, for example, is to generate very detailed maps for a specific time period, telling us how many Zika infections there were in specific places in the world. And we can also, since we are tracking traveling within our simulations, account for how many new cases of Zika are imported because of air traveling to different countries, and then compare those data with the ones that are actually observed and collected by local authorities, like, for example, the CDC in this case. So what's next for us? Well, as I told you at the beginning, our platform now can, of course, model Zika virus, but in principle can be used and extended to study also other vector-borne diseases. For example, dengue, yellow fever, and chikungunya. Therefore, please stay tuned to hear more from us in the future regarding this. But if in the meantime you want to know more about all our other projects and all the amazing people that actually worked on this project, please have a look at our website where you can find more information about this project and all the other amazing things we have been doing. And thanking you for your attention. I want to leave the floor to Salvador to tell you about how you can simulate the brain with GCP. Thank you.
Hi, everyone. So I want to start by quoting President Obama. 2 years ago, he was surprised that as human beings, we were able to understand particles, subatomic particles. We were able to understand galaxies light years away, but we were still unable to understand the brain, the mysteries of the brain. That 3 pounds of matter between our ears, which is basically responsible for everything that we experience and everything that we do. He knew that this knowledge could be transformative, so he decided to start a massive project, research project, called the Brain Initiative, which is over $4 billion, to try to understand the brain. Many other countries in the world have done similar things, and that's why understanding the brain is now considered the new space race. So today I want to talk about 3 things. First, why study the brain? Why is it important? How we can use simulations to understand the brain, and how will cloud help to run these simulations. So why study the brain? The main motivation for me was knowing that 1 out of 6 people suffers a brain disorder. These range from depression, anxiety, Parkinson's, epilepsy, schizophrenia. So this means that if you count 5 people in your row, 1 of you is likely to have one of these disorders. As an example, I want to show you what we can do to help with these disorders. So I'm going to show you a video from CBS 60 Minutes magazine, which shows a woman who's paralyzed from neck downwards. So she cannot move any part of her body, but she has an electrode implanted in her brain. So with this electrode, we can read the signals of the motor cortex, which controls the movements. And then she's able to move an arm almost as if she wasn't paralyzed. There's no sound, but she's basically explaining that she can move the arm up and down. She can close the fist, open the fist, and she can now even try to shake the hand of the presenter. The presenter is pretty amazed that this can happen, and now she offers to do a fist bump. So this is obviously pretty amazing. And the reason this can happen is that we actually understand how some of the signals in the brain are encoded, and we can use that information to decode them. But of course, there are many things that we can do now to improve this. As you saw, the movements are not yet very fluent, very smooth. So there's many things that we can do to understand the brain better. So how do simulations help understanding the brain? First, I want to start explaining why the brain is so complex to understand. And the reason is that there are many levels which are nested inside each other. So this could be like an analogy to nested Russian dolls. So if you start from the higher level, you see that this outer layer called the cortex is sort of, if you stretch it out, it would be like the size of a pizza. And it has many different regions which have different functions. So for example, to process vision, to move your body. So if you go and zoom in into one of these regions, what you see is a very complex circuit of neurons. They are also structured in different layers. There's 6 layers of neurons, and they process all the information, encode what you're doing, And if you go even and zoom in into one of these single neurons, you would see that this is also a very complex machine. So it has lots of chemical processes going on, lots of electrical activity. And what's interesting and complicated is that these 3 different levels interact with each other. So if you want to test something at the molecular level, so you, for example, try an antidepressant drug, an antiepileptic drug, caffeine, you have to check what happens at the single cell, single neuron level. That's going to affect the circuit level, then in turn the whole brain oscillations, for example, and finally the behavior of the human being. So how can we make sense of all this complexity? So luckily for us, there's been many years now of experiments done on the brain, both measuring the shape of neurons, how they're connected, the function of the neurons. And what we can do with simulation is try to put all this together. So here's an example of a simulation based on lots of experimental data from many years. And what we do is reproduce as accurately as possible the experimental data that we have. And then we can use these models, which are very similar to the real brain, to test, experiment with, for example, new treatments to treat some of these disorders that we saw. The advantage of having a simulation is basically that you can test as many combinations as you need. So in the real brain, it's very hard to test different treatments because of ethics reasons. So in the simulation, you can do thousands of different tests with different parameters, different combinations. So I'm going to show you an example of some of the modeling that I do. It's centered in the motor cortex, the part of the brain that controls movements. And here I'm showing the basic level. So this is a single neuron from the motor cortex. And it's important to model very accurately the single neurons because they are basically the building blocks of the brain. So a single neuron can be responsible for a perception, can be responsible for moving an arm. They integrate information from lots of other neurons and then perform functions. The shape is a very important aspect. So if you have the wrong shape in a neuron, for example, less branches, that could lead to disorders like Alzheimer's. Or if you have a neuron that responds too much electrically, that could lead to epilepsy. So it's important to model very accurately both the shape, as we see here. On the left, we have the experimental data. That's an image from 3D microscopy. And then the simulation, which is very similar. And also the electrical response. So we have the experimental data, and we have the simulation, which is, again, very similar to the original. So to simulate these neurons, we use a very detailed mathematical model, which requires solving differential equations, hundreds of differential equations every millisecond. And it basically calculates the currents and the voltages at every point of the neuron. So this was an example for a single neuron, but what I do is simulate a circuit. So I simulate a brain tissue of around the size of a grain of salt, which includes 10,000 neurons, as detailed as the one I showed before, and over 30 million connections. So these connections are based on experimental data. They depend on the cell type, on the layers where they are positioned. They have inputs from other regions. And using this model, we can now check how information is processed, encoded in these circuits, and we can also test all these different drugs and treatments. So as an example of how much computing power this requires, 1 second of this simulation with 10,000 neurons and 30 million connections requires around 1 hour in 50 cores of the supercomputer. So how did I use Google Cloud to simulate the brain? So I mentioned one of these simulations required 50 cores, but of course we need to run thousands of these simulations. Why? First, because we need to optimize the model to reproduce the real brain biology. So we have to run many parameter combinations to check that our model actually does what the real brain does. And secondly, once we have a model which is actually close to the real brain, we can use it to experiment with it. We can, for example, test all these treatments, test also electrical stimulation treatments, which is a new type of method that is used to cure disease. And so we need to run thousands of these simulations. So as an example, if I wanted to run 1,000 of these 1 second simulations, I would require 50,000 core hours. 1 hour in 50,000 cores. So this is a massive amount of computing power that is required, and I want to show you the evolution of how I started using supercomputers. We started with a university cluster which had 500 cores, and this very quickly showed it was very limited. We then used an NSF scientific shared cluster with around 7,000 cores, but it also turned out to be not enough for our simulations. And we have very recently moved to Google Cloud, which allows us to use up to 50,000 cores at the moment, but you can actually use more. And this translates, just as an example, to show you the amount of time required to run these 1,000 brain simulations. So in the regional university cluster, that would be over 120 hours. In the scientific shared cluster, it would be over 30 hours. And in Google Cloud, I can just use 1 hour to run all the simulations. So this, for me, means that I can speed up my research enormously. I can think of an idea, a treatment, design the simulation, and test them in just 1 hour, which is really helpful for me. So I just want to show you some of the results, illustrating some of the results obtained in these simulations. So here we see in the background a representation of this motor cortex circuit. And we see on the top left the diffusion of calcium, which is one of the molecules used in synaptic plasticity. So it's involved in learning, in Alzheimer's, and we can see how it diffuses around the neuron. We can also see the electrical activity, the spikes of the neuron at the bottom, which is the way that neurons communicate. And we can see two measures of the statistics of how populations fire and the oscillations of these populations. These are related, for example, to things like epilepsy. So what are some of the advantages of using Google Cloud, in particular compute engines, which is what I used? As I mentioned already, I have virtually unlimited number of nodes that I can use, and I have no queues. In most of the other supercomputers, there are queues of over 1 or 2 days before the jobs actually start running. The other advantage is I can deploy fully customized clusters, so I can build the cluster to my requirements specifically, which makes it very efficient. And the cluster, importantly, scales up automatically. So as soon as I start submitting jobs, new nodes start lighting up, and once the job's finished, they turn off. So that makes it very efficient and very cost-effective. It is also integrated with the SLARM workload manager, which allows you to submit jobs very easily. It allows to have several users connect to the supercomputer and submit jobs. And it allowed me to reuse some of the code, the scripts that I had for previously tools that I used using SLARM. So I could just deploy them and use them directly in Google Cloud. Finally, I'm also taking advantage of these preemptible nodes, which are basically 4 times cheaper. The disadvantage is that they get preempted, so they are taken away around 10% of the time. So to minimize the impact of getting preempted, we learned a couple of tricks. In particular, if you balance the duration of the simulation and the number of cores you use, by using less cores, you can reduce the chance of these nodes getting preempted. You can also run during the night, which is a simple trick, and it means that less of the jobs get preempted. And you can also generate scripts that automatically resubmit the jobs so that you don't have to take into account whether some jobs finish or not, and they are just, after some time, they will all be completed. So I just want to finish by concluding that Google Cloud is basically helping us understand and treat brain disorders. As I mentioned before, these brain disorders affect a large part of the population. And so there are many promising lines of research now that are happening. And this space race for understanding the brain is just beginning. So I think we can all look forward to very, very exciting times ahead. Thank you. And I want to pass to Ben, who's going to be talking about investigating microbes.
Thank you. Hi, everyone. I want to start my talk with this quote. Microbes run the world. More than half of the oxygen we are breathing is generated by microbes. More than half of our body is microbe. And studying the microbial ecosystem are key to our survival. One of the methods to study microbial life is metagenomics. Metagenomics, we go and take a sample from the environment and trying to understand who is there and what they're doing. And this gives us a snapshot we can try to gather and understand what's happening in that environment. And our goal is to collect as many snapshots as possible to be able to hopefully make a climate change model. And then we see how the microbial life changing as climate changing, because that's going to be key for us. In our study, we actually go to the island in Bahamas called San Salvador. And you can see these islands are really unique because they have many, many ponds. And you can see the study pond, the one we are doing our work, is marked with the red arrow right there. They're unique because they're close to the ocean, but they're closed pond. They're not open to the water. They go through much rapid climate change compared to any other environment. That enables us to observe the climate change much faster. And when we take a sample, we can have a better understanding how the climate change affects our sample. And we're trying to kind of collect all this information whenever we take in any sample from these environments. And I'm sure, same Bahamas, that's what you guys thinking. Yes, it's beautiful. We really have fun to go in there. But most time, this is our view. Because these ponds are so close to environment, they go through really harsh environment. And in this pond, you guys see the salinity is really high. The water changed yellow. We saw even purple or other colors. Depending on what happened to that environment, the color of water changing. And that's really interests us to see what happened to those microbial ecosystem in that environment. And as mentioned, we go there literally trying to not disturb the environment and trying to pick up the sample, then take the sample to our lab, trying to sequence it and run our computation to understanding what's going on in our sample. But still we're dealing with two main issues. One of them are data getting bigger and bigger. I'm sure everybody here heard the sequencing technology getting cheaper and cheaper. We're sequencing our own DNA, our dogs now, and we're getting more and more data. And we're dealing in our experiments for each snapshot like 10 terabytes of data. For the study we are working on, we have 10 snapshots. We're dealing with 100 terabytes of data to compute. And that's a lot of data to just go through. Also, if you guys know bioinformatics or genetics algorithm, they're computationally expensive. Everybody, I'm sure, heard about assembly or alignment. There have been a lot of optimization to these algorithms, but still they're heavily computationally expensive, and there's not much we can do but just compute the data. As data get growing, the compute time growing too. What we try to do, we come back and look at our workflow in a lab, and we understand the compute time and analytic part is the main part we need to focus and trying to solve the issue. And that was we kind of trying to go our university cluster and see what we can do and change to our workflow to be able to compute our data much faster. We come up with our own workflow. We design our own workflow to be able to scale up our job. But if you're familiar with the workflows, the second you see this picture, you should recall this. This is MapReduce. What we did, we adopt the MapReduce model to our workflow. And you can see in the top, you can mention all the parts we are doing to our workflow is in the top, and you can see how it's matched to the MapReduce. And that was the reason we thought, oh, we solved the issue. Now we can scale as much we need. But in reality, when we run our job, we were really happy to see the job was taking 7 years to change to 3.5 months. But if you know research, most time we have 1 or 2 years. If you get a life grant, it's 5 years. And I mentioned we have 10 snapshots. In this case, we're going to be around 3 months just computing the data. I don't know when we're going to be able to actually publish or do the work. That was the reason we kind of reached to our friend at Google and asked them for help. And we estimate after we transition to GCP, we can finish the same work in 12 hours. That's a big deal. I'm going to invite here my friend Paul to tell you guys how we move our workflow from our local cluster to GCP. Thank you.
Hello. All right. That's right. Thanks, Ben. Yeah, so as part of the HPC team at the university, our job is to help them accelerate their research. And if we see that we can go from 7 years to half a day, well, we need to make that happen. So our first step was to come up with a plan. We really wanted to deploy a cluster in the cloud that happened to look like our environment back home. We wanted to dynamically scale that environment based on his job size or future job sizes. We also wanted to dynamically install a software stack based on the software he needed or needed to change and so on. We also had to come up with the best storage solution because we're dealing with a lot of data and one mistake could cost us a lot. So next stage was doing small tests. You know, we needed to iterate through the process to basically optimize, make sure that we're doing this exactly right, or else, again, it could cost us. We had to develop tools to basically script out automated job submission because of the large set of jobs and samples that we had to work with. After we did that, we were able to scale up to smaller just to make sure our scaling is working right before we have to send out that big job. And then lastly, that big job. We have to hit the go button. Submit the entire work set, monitor, and verify that these results are coming back right. So this is what our solution ended up looking like. And you know, you can kind of think of this as our input, our compute, and our output. Now our input is our reference database, which is actually going to be the same for every job that we run. And then of course our sample database. We ended up using an attached persistent SSD for this entire run. Every job had this attached persistent SSD, but the key was it was read-only. This worked extremely well for us. It was fast and scaled extremely well. Our compute. So our compute is our dynamic HPC cluster, and as I said, I wanted this to look like our cluster back home. So it does. We have a login node, a controller node, and our compute stack. Our compute stack ended up being the 32 core instances out there. Now this is all managed with the Slurm workload manager. So as mentioned in the previous talk, our jobs actually scaled with the cluster. So it worked out great. Our output. So our outputs are actually relatively small. And you know, we didn't really need big scale SSDs, big scale file storage. We actually sent our data at the end of our jobs to a Google Cloud bucket. So the job. We ran on 124,352 cores concurrently. We ran on 3,886 nodes, and we did that in 16 and a half hours. So challenges and findings for our run. It was enormously successful. I think we had 2 instances where the nodes didn't come up right. We resubmitted those jobs automatically through our scheduler. It worked great. During the testing phase, as I mentioned, we had to find that best storage. We tried NFS, we tried one of the Elastifile, which was neat, but kind of overkill, and tried some local disk options. And you know, we ended up going with that combination of the local SSD and connected to that persistent SSD. Worked great. Slurm. So Slurm was a lot of fun. You know, we used the Slurm workload manager back home on our cluster, so we were already very familiar. Working with the Google team and the Slurm team, we went through a lot of tweaking, a lot of making it just how we wanted. Lastly, something we didn't anticipate is hyperthreading. In our HPC space, in most HPC shops, we don't use hyperthreading. It just doesn't provide any advantage. It's already enabled in the cloud, so that actually affected our runtime slightly. So it's just something for us to keep in mind in the future. So our takeaway and our key next steps, this greatly enhanced our research. I mean, it really made this a reality for this lab. Our collaborative process with the Google team and the Slurm SchedgeMD team was fantastic. You know, we provided feedback, they provide suggestions. I think we actually led to some code changes and updates and things like that. My goal even before this and still moving forward is us to locally burst our environment into cloud. And this is exactly what it was. So we're really looking forward to the future. And now that this particular one has been a success for this research lab, that lab is now able to seek further funding and able to expand the research. You know, climate change doesn't happen in a day. He needs samples for months, years, even decades. So he already has about a year's worth of data ready to compute, and if he tried to continue what he was doing on his local PC, it would take him 50 years just for a year's worth of data. On our cluster, 2.2 years, and that's if I actually let him on my cluster for 2.2 years dedicated. GCP, 3.5 days. I'd like to thank Bob Doran. He's another member of our group that made this project possible. Sean Norman, who is the lead PI for the research, and of course our Google team.